The evolution of microbial life. Our planet's history began about 4.6 billion years ago. 3.5 billion years ago, the first living organisms appeared. The earliest organisms were all prokaryotic. Prokaryotic organisms are single-celled life forms that lack membrane-bound nuclei and lack membrane-bound organelles. The early atmosphere lacked oxygen. 2.7 billion years ago, atmospheric oxygen began to accumulate as a byproduct of the photosynthesis of autotrophic prokaryotes. The first eukaryotic organisms appeared about 1.8 billion years ago. Eukaryotic life forms can be single-celled or multicellular. The cells of eukaryotes contain nuclei and other membrane-bound organelles. The first eukaryotic organisms to appear on Earth were the protists. Protists are mostly microscopic and unicellular, and they are represented today by a great diversity of different species. Eukaryotic cells most likely evolved from ancestral host cells that engulfed smaller prokaryotes. This idea is called the endosymbiotic theory. The mitochondria of our cells and those of every other eukaryote are descendants of those prokaryotes. The same is true of the chloroplasts found in plants and algae. The oldest fossils that clearly showed multicellular organisms are about 1.2 billion years old. The first multicellular organisms were simple and very small. Large, diverse multicellular organisms did not appear in the fossil record until about 635 million years ago. The Cambrian explosion began about 541 million years ago and marked the beginning of the Paleozoic era. In the Paleozoic era, about 500 million years ago, the dry land on Earth began to be colonized by plants, fungi, and insects. The Mesozoic era is sometimes called the Age of the Dinosaurs. During the Mesozoic era, more and more familiar organisms begin to dominate the landscape, including flowering plants, birds, and mammals. Modern humans are Homo sapiens. We appeared roughly 195,000 years ago. Early in its 4.6 billion-year history, our planet had no lakes, no oceans, and very little oxygen. At this time, Earth was bombarded by debris that generated heat. This vaporized all available water. About 4 billion years ago, the water vapor had condensed into oceans. The surface cooled. Volcanic eruptions continued, adding carbon dioxide, methane, ammonia, and other nitrogenous compounds into the atmosphere. One of the more popular hypotheses for the origin of life is called the four-stage hypothesis. The four-stage hypothesis explains how the first organisms were likely products of chemical evolution that occurred in 4 stages. The first stage involves the synthesis of small organic molecules, such as amino acids and nucleotide monomers. The second stage involves joining these small molecules to form macromolecules, including proteins and nucleic acids. The third stage is the packaging of all these molecules into precells. These precells are essentially liquid droplets with membranes that maintained an internal chemistry different from its surroundings. The fourth and final stage is the origin of self-replicating molecules that eventually made inheritance of genetic material possible. Let's look at each of these stages more closely. In stage 1, we have the synthesis of organic compounds. The chemicals that formed on Earth just after its formation included water, methane, ammonia, and other small inorganic molecules. However, the complex organic molecules needed for life, including sugars, fatty acids, amino acids, and nucleotides, are composed of these same elements. But the question remained, could these complex molecules have arisen from these small inorganic molecules? In 1953, Miller and Urey performed an experiment. They created an apparatus that would simulate the conditions of the early atmosphere of Earth. They included an atmosphere containing hydrogen gas, methane, ammonia, and water vapor. They included a flask of warmed water to simulate the primordial sea. After about 1 week, the apparatus contained an abundance of organic molecules essential for life. These included amino acids, which are the monomers of proteins. These molecules had shown up in the water of the apparatus that was representing the primordial sea. There are a lot of different theories about where life began. These theories include submerged volcanoes, deep-sea hydrothermal vents, and meteorites. Meteorites contain some organic molecules including amino acids, lipids, simple sugars, and even nitrogenous bases. In stage 2, we have the abiotic synthesis of polymers. Once small organic molecules were present on Earth, how is it possible that they were linked together to form polymers, such as proteins and nucleic acids, without the help of enzymes or other cellular equipment? It has been demonstrated in the laboratory that such polymerization can be brought about by dripping solutions of organic monomers onto hot sand, clay, or rock. The heat vaporizes the water in the solution and concentrates the monomers on the underlying material. Some of these monomers then became spontaneously bound to one another, forming polymers. On early Earth, raindrops or waves may have done the same thing, concentrating organic monomers onto fresh lava or other heated rocks, allowing polymers to form. Stage 3 is the formation of pre-cells. An important step in the origin of life would have been the isolation of a collection of organic molecules within a membrane. These molecular aggregates are called pre-cells. Researchers have demonstrated that pre-cells could have formed spontaneously from fatty acids. The first membranes were likely quite porous. This would allow organic monomers, such as RNA nucleotides and amino acids, to cross freely. However, polymers that formed inside were too large to exit. They may have had the ability to use chemical energy and grow. Pre-cells made in laboratory experiments have been shown to divide, producing new pre-cells in a simple form of reproduction. Stage 4 involves the origin of self-replicating molecules. What were the first genes like? One hypothesis is that the first self-replicating molecules were short strands of RNA. These short RNA strands replicated themselves without the assistance of enzymes. Laboratory experiments have shown that short RNA molecules can assemble spontaneously from nucleotide monomers. When RNA is added to a solution containing a supply of RNA monomers, new RNA molecules assemble, using the previous RNA molecules as a template. Some RNA monomers spontaneously join, forming the first small genes. Then, an RNA chain that is complementary to one of those genes assembles. If the new chain then serves as a template for another round of RNA assembly, the result is a replica of the original gene. Each pre-cell would contain its own population of RNA molecules, each with a random sequence of monomers. Some of these molecules would self-replicate, but their reproductive success would vary. Pre-cells containing more advantageous genes that help them survive and reproduce then increased in number, passing their abilities on to later generations. Mutations would have resulted in additional variations on which natural selection could work. The most successful pre-cells would have continued to survive, reproduce, and evolve. At some point during millions of years of selection, DNA, which is a more stable molecule, replaced RNA as the inherited genetic material. These protocells then evolved through several unknown steps to become true cells. The human microbiota is the community of microorganisms that live in and on our bodies. Some of our intestinal inhabitants supply us with essential vitamins. They also enable us to extract nutrition from food. Many of the bacteria that live on our skin perform helpful housekeeping functions, such as decomposing dead skin cells. Prokaryotes also guard the body against disease-causing microbes. Prokaryotes living in the soil and at the bottom of lakes, rivers, and oceans help to decompose dead organisms and other waste materials. This recycles crucial chemical elements, such as nitrogen, and places them back into the environment. Without prokaryotes, the cycles that sustain life would come to a halt, and all forms of eukaryotic life would no longer be possible. In contrast, prokaryotic life would persist in the absence of eukaryotes, just as it did for 2 billion years on early Earth. Prokaryotic organisms come in many different forms. Determining cell shape by microscopic examination is an important step in identifying a prokaryote. The three most common shapes of prokaryotic organisms are spherical, rod-shaped, and spiral. Round prokaryotic cells are called coccus, or cocci for plural. Rod-shaped prokaryotic organisms are called bacillus, or bacilli for the plural form. Spiral-shaped prokaryotes include the spirochetes, which include different species that cause syphilis and Lyme disease. Although all prokaryotes are unicellular, the cells of some species usually exist as groups of 2 or more cells. Cocci that occur in clusters are called staphylococci. Cocci that occur in chains are called streptococci. These include the bacterium that causes strep throat. Some prokaryotes grow as branching chains of cells. Some species even exhibit a simple division of labor among specialized types of cells. There are even some giant prokaryotic cells that are much larger than some eukaryotic cells. The giant bacterium, which lives in the gut of surgeonfish, is large enough to be seen with the naked eye and can grow up to 10 times the size of a typical eukaryotic cell. In many natural environments, prokaryotes attach to surfaces in a highly organized colony called a biofilm. A biofilm may consist of 1 or more species of prokaryotes, and it may also include protists and fungi as well. As a biofilm becomes larger and more complex, it develops into a community of microbes. The cells within this community communicate to each other using chemical signals. Members of the community enjoy a division of labor. Biofilms can form on almost any type of surface, including rocks, organic material, including living tissue, metal, and plastics. Many of the bacteria that cause disease in humans are capable of forming biofilms. In fact, biofilms are often the cause for dental plaque, ear infections, and urinary tract infections. The structure of biofilms makes these infections exceptionally difficult to treat. Antibiotics may not be able to penetrate beyond the outer layer of cells, leaving much of the inner community unharmed. Binary fission is the process by which most prokaryotic cells reproduce. Some species can produce a new generation in just 20 minutes under optimal conditions. If reproduction continued unchecked at this rate, a single prokaryote could give rise to an entire colony that outweighs the Earth in only 3 days. The DNA gets replicated for each round of binary fission. It is here when spontaneous mutations can occur. These mutations can generate genetic variations in prokaryotic populations. When the environment changes, only individuals having beneficial genes survive. Such genes would enable the individual to quickly take advantage of the new conditions. So, why are we not knee-deep in prokaryotic organisms? Well, several factors limit prokaryotic cell division. These include limited resources, such as food and space, and a buildup of metabolic waste products that eventually pollutes the colony's own environment. Some prokaryotes can survive during very harsh conditions by forming specialized cells called endospores. An endospore is a thick-coated protective sphere that forms inside of the prokaryotic cell. The formation of the endospore protects the prokaryote when it is exposed to unfavorable conditions. Endospores allow prokaryotes to survive such things as trauma and extreme temperatures. When the environment becomes more hospitable, the endospore can absorb water and resume growth. The prokaryotic pathways that transform energy and matter are far more diverse than those in eukaryotes. Some species harvest energy from inorganic substances such as ammonia and hydrogen sulfide. Some bacteria found in soil are able to obtain energy from inorganic nitrogen compounds. This bacteria is essential to the chemical cycle that makes nitrogen available to plants. Some bacteria use sulfur compounds as a source of energy. These bacteria support diverse animal communities. Prokaryotes can thrive in conditions that seem totally inhospitable to life. Prokaryotes have been found between layers of rock buried hundreds of feet below the Earth's surface. They've also been found near deep-sea hydrothermal vents, where scalding water and hot gases surge into the water. Some prokaryotes are considered symbiotic. Symbiosis is a close association between organisms of 2 or more species. For example, many of the animals that inhabit hydrothermal vent communities have sulfur bacteria within their bodies. The animals absorb sulfur compounds from the water, and the bacteria uses these compounds as an energy source. The bacteria converts these compounds into organic molecules, which then provides food for their hosts. Nitrogen fixation is the process of converting atmospheric nitrogen into a form usable by plants. Symbiosis with cyanobacteria gives plants an advantage in nitrogen-poor environments. Other nitrogen-fixing bacteria live symbiotically in root nodules of plants in the legume family. Life depends on the recycling of chemical elements between biological and physical components of the ecosystem. Nearly all the nitrogen that plants use comes from prokaryotic metabolism occurring in the soil. In turn, animals get their nitrogen compounds from the plants. Prokaryotes decompose organic matter and return vital elements to the environment that can then be used by other organisms. Without these decomposers, carbon, nitrogen, and other elements essential to life would become locked in the organic molecules of corpses and waste products. Bioremediation is the practice of using organisms to remove pollutants from water, air, and soil. Prokaryotic decomposers can also be used to treat sewage. Prokaryotes decompose the organic matter in the sludge, converting it to material that can then be used as fertilizer or landfill. Aerobic prokaryotes and fungi remove most of the organic matter from the sewage. The outflow is then sterilized and released back into the environment. Bioremediation has also become an important tool for cleaning up toxic chemicals released into the soil and water by industrial processes. Many prokaryotes are capable of decomposing pollutants such as oil, solvents, and pesticides. In 2016, researchers discovered bacteria that can metabolize plastic. There are two major branches of prokaryotic evolution: bacteria and archaea. Archaea are abundant in many habitats, including places where few other organisms can survive. The extreme thermophiles, or heat lovers, live in very hot water, sometimes near boiling. The extreme halophiles, or salt lovers, are found in areas like Utah's Great Salt Lake and the Dead Sea. They are also found in seawater evaporating ponds that are used to produce salt. One group of archaea are the methanogens. Methanogens live in anaerobic or oxygen-free environments. They produce methane as a waste product. Methanogens are found in solid waste landfills and in the mud lying at the bottom of lakes and swamps. Large numbers of methanogens also inhabit the digestive tract of animals. In humans, intestinal gas is largely the result of methanogen metabolism. Methanogens aid digestion in cattle, deer, and other animals that depend heavily on cellulose for their nutrition. Pathogens are infectious agents or bacteria that cause disease. Endotoxins are chemical components of the outer membrane of certain bacteria. All endotoxins cause the same general symptoms: fever, aches, and a dangerous drop in blood pressure, which can lead to septic shock. Septic shock can kill a healthy person in a matter of days or even hours. Examples of endotoxin-producing bacteria include the species of Salmonella that causes food poisoning and typhoid fever. Staphylococcus aureus is the leading cause of food poisoning. Neisseria meningitidis is the bacteria that causes meningitis. One of the greatest threats is from endospores of the bacterium that causes anthrax. When anthrax endospores enter the lungs, they germinate, and the bacteria multiply, producing an endotoxin that eventually accumulates to lethal levels in the blood. In a terrorist incident in 2001, 5 people died when anthrax spores were mailed to members of the news media and to the U.S. Senate. Lyme disease is caused by a spirochete bacterium carried by ticks. Disease-carrying ticks live on deer and field mice, but they can also bite humans. Lyme disease usually starts as a red rash shaped like a bullseye around the tick bite. Antibiotics can cure the disease if it is administered within a month of exposure. However, if left untreated, Lyme disease can cause debilitating arthritis, heart disease, and nervous system disorders. Since there is no vaccine, the best defense against Lyme disease is public education about avoiding tick bites and the importance of seeking treatment if a rash develops. Unlike other biological agents, the weapon of Clostridium botulinum is an exotoxin that produces botulinum. Botulinum is the deadliest poison on Earth. It blocks transmission of the nerve signals that cause muscle contraction. Botulinum results in paralysis of the muscles required for breathing. 30 grams of botulinum, which is a bit more than an ounce, is enough to kill every person in the United States. The term protists is not a taxonomic category. Recent genetic and structural studies shattered the notion of protists as a unified group. Some protists are more closely related to fungi, plants, or animals than they are to each other. Protists is a catch-all category for all eukaryotes that are not fungi, animals, or plants. Most, but not all protists are unicellular. However, all protists are considered eukaryotic. Photosynthetic protists belong to an informal category called algae, which also includes cyanobacteria. Algae may be unicellular, colonial, or multicellular. Other protists are heterotrophs that eat other organisms. Some heterotrophic protists eat bacteria or other protists. Some obtain organic molecules by absorption, while others are parasitic. A parasite obtains its own nutrition from a living host, which is often harmed by the interaction. For example, the trypanosomes are parasites that attack human red blood cells, causing sleeping sickness. Sleeping sickness is a debilitating disease common in 2 parts of Africa. Other protists are mixotrophs, which are capable of both photosynthesis and heterotrophy. Euglena is a common inhabitant of pond water. It can change its mode of nutrition depending on the availability of light and nutrients. Protozoans are protists that live primarily by ingesting organic matter. Protozoans thrive in all types of aquatic environments. Most species eat bacteria or other protozoa. Some are even able to simply absorb the nutrients dissolved in water. Protozoa that live as parasites in animals, though in the minority, cause some of the world's most harmful diseases. Flagellates are protozoans that move by means of one or more flagella. Most flagellate species are free-living and non-parasitic. However, this group also includes some harmful parasites that make people sick. One example is Giardia. Giardia is a common waterborne parasite that can cause severe diarrhea. Another flagellate is Trichomonas. Trichomonas is a common sexually transmitted parasite. Trichomonas can also be contracted by ingesting food contaminated with feces containing the parasite. The parasite then travels through the reproductive tract by moving its flagella and undulating part of its membrane. In women, Trichomonas feed on white blood cells and bacteria living in the female reproductive tract. Trichomonas also infects the cells lining the male reproductive tract, but limited availability of food results in a very small population size. Consequently, males typically do not have any symptoms of infection. Other flagellates live symbiotically, in relationships that benefit both partners. Termites are notoriously destructive to wooden structures, but they lack enzymes to digest the tough, complex cellulose molecules that wood has. Flagellates residing in the termite's digestive tract break down the cellulose into simpler molecules. They then share these nutrients with the host. Amoebas are characterized by great flexibility in their body shape and the absence of permanent organelles for locomotion. Most species move and feed by means of a pseudopodium, which is a temporary extension of the cell. One species of parasitic amoeba causes amoebic dysentery. This disease is responsible for an estimated 100,000 deaths worldwide every year. Other protozoans with pseudopodia include the forams, which have shells. Although they are single-celled, the largest forams can grow to a diameter of several centimeters. 90% of forams that have been identified are fossils. Apicomplexans are all parasitic, and some cause serious human disease. They are named for a structure at their apex, or tip, that is specialized for penetrating the host's cells. Apicomplexans include Plasmodium and Toxoplasma. Plasmodium is the parasite that causes malaria. Toxoplasma gets transmitted by handling the litter of outside cats. A woman who is newly infected with Toxoplasma during pregnancy can pass the parasite to her unborn child, who may suffer damage to the nervous system as a result of the infection. Ciliates are protozoa named for their hair-like structures called cilia. These cilia allow the protist to be mobile. They are also used to sweep food into the protist's mouth. Nearly all ciliates are free-living or non-parasitic and include both heterotrophs and mixotrophs. A paramecium is a great example of ciliate. Slime molds are multicellular protists related to amoebas. Slime molds were once classified as a fungi But DNA analysis showed that they arose from different evolutionary lineages. Like many fungi, slime molds feed on dead plant material. There are two distinct types of slime molds: cellular slime molds and plasmodial slime molds. Cellular slime molds exist as separate individuals for most of their life cycle. The cellular slime molds only come together to form a mass when conditions require it, such as when food is scarce. In contrast, plasmodial slime molds form a large, multinucleated mass of protoplasm, essentially acting as a single giant cell, where many nuclei share a single plasma membrane. Many unicellular algae are components of phytoplankton. Phytoplankton are the photosynthetic organisms that are mostly microscopic and drift near the surfaces of ponds, lakes, and oceans. Dinoflagellates are abundant in the vast aquatic pastures of phytoplankton. Each dinoflagellate species has a characteristic shape, reinforced by external plates made up of cellulose. The beating of two flagella, located in perpendicular grooves, produces a spinning motion. Population booms of dinoflagellates sometimes cause warm coastal waters to turn pinkish-orange in a phenomenon known as red tide. Some of the dinoflagellates release toxins into the ocean, causing massive fish kills. They can be poisonous to humans as well. One group of dinoflagellates resides within the cells of reef-building corals. Without these dinoflagellates, corals could not build and sustain the massive reefs. These reefs provide food, living space, and shelter to an astonishingly diverse community of organisms. Diatoms have glassy cell walls containing silica. Diatoms store their food reserves in the form of an oil that provides buoyancy. This buoyancy keeps the diatoms floating. We refer to these diatoms as plankton as they approach the water's surface. The organic remains of diatoms that lived hundreds of millions of years ago are thought to be the main component of oil deposits. Green algae are named for their grass-green chloroplasts. Unicellular green algae flourish in most freshwater lakes and ponds, as well as many home pools and aquariums. Colonial forms of green algae include volvox. Each volvox colony is a hollow ball of flagellated cells. The spheres inside the volvox are daughter colonies that will be released when the parent colonies rupture. Seaweeds are classified into three groups, based partly on the types of pigments presented in their chloroplasts. These groups include green algae, red algae, and brown algae, some of which are known as kelp. Thank you for watching.